Okay, excellent. Well, what I'd like to present in the today, start with a little background on biogenic VOC. Talk a bit about what I see are some of today's research challenges in this field, and then focus on one of those research challenges, which is BVOC emissions from managed landscapes, something I think we haven't been doing very well. And so I'll talk about why I think it's important to do that, some of the modeling approaches we're starting to use for this, and also just the implications of having good BVOC emission estimates in managed landscapes. Well, first of all, what are these BVOC? Well, these are, for those who aren't familiar, these are scents, many of these are scents that you do know as you walk outside through a forest, as you sit down to eat or have a beer. These are a lot of familiar odors. Some of them, though, are not odors that our nose is particularly good at picking up. Isoprene, for example, is the globally dominant BVOC, but it doesn't have much of a smell, so you're not real familiar with it. Another thing about these is that a lot of these compounds, we don't recognize individual odors. A lot of things like mushrooms and roses and oranges will have a blend of different compounds. So one thing that we're, as scientists, often curious about are why are organisms making all these BVOC? And globally, plants are the biggest source of these VOC. And we understand some of the reasons that they do, part of pollination and disseminating seeds, protection from herbivores and predators, protection from abiotic, heat stress, drought stress. But there are others that we don't really understand. Many of them we don't fully understand the role of these compounds. For atmospheric chemists, the interest here is, well, what is the impact of this huge amount of reactive organics going into the atmosphere? And in the clean atmosphere, mostly these BVOC just get chewed up to carbon dioxide and other small compounds that get redeposited to the Earth's surface. Now, in that process of getting chewed up, they're also using up a lot of oxidants in the atmosphere. And so that's their primary impact in the clean atmosphere, is to reduce the amount of oxidants, particularly the OH radical. But in a polluted atmosphere, it's quite different. These compounds, these BVOC, end up producing, in some cases destroying, since they do destroy oxidants, but they also can end up to a production of ozone and particles. And these are two of the most important regional air pollutants, and also they're short-term climate forces. Now, we've had an understanding of this going back to the 1960s and 1970s, particularly pioneering work by Professor Fritz Wendt and Ry Rasmussen, and both their impacts on particles in the atmosphere and ozone. Now, we've typically thought that that's 60s and 70s is when this research started, but we'd actually learned that this work was going on in the then Soviet Union. Back to the 1920s into the 1950s, there was a lot of active research going on there, which just points out the importance of having organizations like iLEAPS, which of course didn't exist back then. And so different communities didn't really know what other communities were doing. And we can't just assume that one group are the only ones working on things. Well, in the key efforts in the 1970s and early 1980s, we're focused on these air pollution issues and just trying to get some general estimates of what was going on, making some initial emission inventories. So people were going around with enclosures, trying to see what different plants were emitting, looking at kind of the simple diurnal variations. And so we learned a lot during this period and came up with some convincing evidence that, that these emissions were real and that they were quite large. But in the 1980s, 1990s, there were questions, though, about what really were these impacts. The concentrations in the atmosphere were low, and some people thought they really weren't very important. But through the work of scientists, and especially the late 1980s, by the early 1990s, it was clear that that these were important and that you weren't going to solve the regional ozone problem. And then later we learned also for particle problems if we didn't understand the biogenic VOC. And this was a time when these VOC started to be included in numerical models of regional air quality and global atmospheric chemistry and climate. And I think particularly impactful was this 1991 National Research Council book that basically said, no, it's the combination of the biogenic VOC and the anthropogenic NOx that makes ozone, and we need to understand this. So as time went on, the 2000s, there was a lot of interest in interactions. And these BVOC are particularly sensitive to changes in land cover, changes in temperature, extreme events, more so than, for example, photosynthesis is. They're more sensitive. And so there was a lot of interest. Well, what kind of feedbacks, since the atmospheric system is sensitive to the BVOC sensitive to the system, you can draw lots of different interactions. And it's easy to conceptualize these, but actually really difficult to try to quantify them and observe them on regional scales. So we've kind of drawn a lot of these cartoons, have the general idea what can go on, and you can run these in models. You can see these interactions in coupled models, but there are so many uncertainties that often we're not even sure of what direction the interactions are going in. In the 2010s, a lot of interest in plant stress, and especially stress is being driven by a changing climate, by changing land use, and a lot of studies working to quantify that, which showed substantial impacts and changes, which again gets back to those interactions that were being looked at. And so clearly these were going on, but again, a lot of uncertainties. Still even difficult to know, what direction these things are going in. And so progress and kind of understanding the general importance, but difficult to get to the details. Still work to be done there. So now in the 2020s, my thoughts on what we are really needing to work on. Still, again, we haven't worked out all those other things, but I think some key things include seasonal to interannual variations and in particular extreme events. We really don't have data sets. We don't have long-term data sets for looking at that. Second, I think with advances in satellite data and low-cost in situ observations, we're really well poised to make some progress in assessing the models that we have. And then finally, managed landscapes that I want to focus on in this talk. But briefly, just talking about those first two that I think are important and key to work on. With seasonal interannual variations, we have, I know most of you know, we have the FluxNet network of carbon and water and energy fluxes. There's over 1,500 site years of data from hundreds of sites. Most of these sites have 7 years of data, at least 7 years of data. With BVOC fluxes, we've got about 20 site years, from about 65 sites with an average of about 4 months per site. So we're not doing so bad in the BVOC community of getting flux measurements at different sites. What we're bad at is getting long-term measurements. And it's understandable. Setting up BVOC fluxes is much more difficult and expensive than setting up an ERGA to do CO2 and water and energy fluxes. And so in our community, a lot of the studies will just be short-term, apply new techniques, look at a new ecosystem, and run for a few months, if that, and then move on to other things. But to really understand these seasonal to interannual variations, extreme events, be able to capture droughts and things like this, we need to have a lot of these data sets in a lot of different communities. And I encourage those of you interested in this to get involved in this. One of the key things we need to do this are lower cost systems. And so one of the things we've been working on in my group are low cost, but continuous systems. So relaxed eddy accumulation systems or true eddy accumulation systems with online GCs, but that are low cost would make this possible. The longest BVOC that we currently have is 5 growing seasons. That was at the University of Michigan Biological Station. And even with this, we think this data set's been looked at for a number of years, so it's almost 20 years old now. With the first pass look at this said basically, oh yeah, our diurnal curves look pretty good. It follows it pretty well. But as we've started to look a little deeper into this and using some kind of modern techniques like machine learning, we've really been learning a lot from this data set and learning where the model wasn't working very well at some higher temperatures and different values. I wanted to mention this is work being done by my PhD students, B. Kitty Nana Vong and Hui Wang. And so in doing this, we've been able to optimize the algorithms that we're doing. And of course, though, what we need to now are more data sets, more long-term data sets that we can apply and compare to, but we just have so few long-term data sets. But indeed, we can see it improving. We can also mark out, well, now it works really well, except for this group of data and what's going on there in that group of data. And along with that, one of the things and why we need these long-term data sets is to be able to look at things like drought and heat waves, which we're just not going to see if we have a bunch of 2, 3, 4 month data sets in various places that tend to be, during normal conditions. We have a few, but not many. And with the few data sets that we have, we've been able to test new models with drought and heat wave stress. We've been able to separate these a little bit. And the model that we use now considers both the heat stress and the increase in temperature that you get during a drought, because the stomata of leaves close up and the leaves heat up, which actually increases isoprene emission. But then there's a decrease that comes because the impact on photosynthesis eventually drives down those emissions. And so so that's one topic. Another is I think there are a lot of improvements on our ability to be able to assess these emissions. There have been a lot of studies that, for example, looking at satellite data that would say, this model is too high or too low, but the uncertainties in the satellite data were 50% or more, and it made it difficult to do that. There's also, I think, ways that we can do this with in situ low-cost observations, where we can look at this. But for example, with the satellite data, previous formaldehyde measurements, would indicate that the isoprene in the models were way too high in this region in Texas, in central Texas, that I'm showing here on the far left. But on the far left are the tropomi observations. And then you can see the next panel over to the right is the bias corrected tropomi, which changes things quite a bit. And then suddenly this model that seemed to be overestimating emissions is actually underestimating emissions in a lot of areas, or getting it close to correct. And so I think we're finally getting to the point where this satellite data is going to be really useful, and especially satellite data that integrates a lot of other observations so that we can get the chemistry right. So that's the key, going between the satellite formaldehyde and emissions of isoprene is that we get the chemistry right and have the right data to start with. Now, another thing then, as we get more confidence with this satellite data and we get higher resolution satellite data, we can actually start using this to focus in on urban areas. In the past, our BVOC emission model, we focused on very large scale features and using models with quite low resolution. Now, for example, here, this is looking at tropomi formaldehyde to NO2 ratio. And that's a really important ratio in telling us where we'd expect ozone production to be VOC sensitive or NOx sensitive. So if we look at this map right here, and we're seeing on the satellite data, the left one and the middle one are satellite data. The far right one is a model being driven with Megan BVOC emissions. But what you can see is that there's a lot of red area, right? And that's the area out in the rural areas where there's lots of VOC emission. And so it's very red, which means it's just not sensitive to VOC at all. What that tells us is we don't need to get the VOC emissions exactly right in those areas that show up here as really red. It's actually in the areas where there are cities. You see those numbers there, and it's kind of whitish and bluish. Well, those are areas where the VOC matters, that we get that right. Well, where have we focused all of our VOC emission studies efforts? Well, it's in those really red areas, right? We go to the isoprene hotspots because, yeah, we can measure it there. We can do chemistry studies there. There's lots of VOC. But the areas that really matter from a local regulatory point of view are in the cities. This is where it really matters, getting the BVOC number exactly right, because these are the VOC sensitive areas. And so we've been, and others have been looking more at this. And this is a paper led by Shangu, who's a PhD student at UCI, working with Celia Fiola. And in this study, we took the Megan model, did a few initial kinds of tweaks to it, because Megan and every other BVOC model simply do not work well in urban areas, right? They weren't designed to do that, and don't have appropriate inputs to estimate it there. But we did an initial tweaking of trying to improve the inputs there, and came up with estimates that indicate for Los Angeles County, so this area population of 10 million, and in there, the emissions of BVOC and anthropogenic VOC for LA County are about the same. But when you look at ozone formation potential, because the BVOC are more reactive, there's actually considerably more ozone formation potential coming from the BVOC than the AVOC. Of course, this is not just in Los Angeles. This is more and more happening in cities, urban areas throughout the world. As we reduce the anthropogenic VOC, then the biogenic VOC come to dominate more and more. So this wasn't the case 30 years ago, but it certainly is now. So with the latest version of Megan, so Megan 2.1, very much focusing on global, was high resolution. So it could also be used in regional models. So it has a resolution of 1 kilometer, but was not intended to work in urban areas. With Megan 3.2, the intent is to be able to do urban areas. as well as to go to the global scale. Some other differences, more of a focus on stress for the algorithms, more of the stress VOC, BVOC compounds. Megan 2.1 is a net ecosystem model. So it doesn't have the individual components in it, but with Megan 3.2, we do. And a major effort with Megan 3.2 to have more transparent emission factors. With Megan 2.1, you can't, there's no indication of where the emission factors come from and how you would adjust them. And we're trying to make that easy for people to add these emission factors in Megan 3.2. So just kind of briefly what those things are. So Megan 2, whole ecosystem, Megan 3 have these various components where we want to be able to model them individually, and with the idea that air quality models are advancing to the point where they can actually have a canopy airspace and do the chemistry there. So having the emissions from the different components and mixing in the canopy, we think is important. So the next challenge with getting BVOC emissions is the initial challenge is just how much of the source is there. So mostly it's from foliage. We do have these other sources as well, but foliage is still by far the dominant. And so in terms of how much foliage, well, we've had LAI data, leaf area index data for a long time, global data sets. This has allowed us to have global 8-day values at 1 kilometer, which really works well for getting regional to global sorts of things. When you get down to urban areas, though, it doesn't work. And a lot of these data sets, some of these LAI data sets actually mask out urban areas. But in any case, they don't do a very good job there. And we've looked at, have been using data sets up to 30 meters, but even then, it doesn't quite get things right. Now, LAI data is available globally throughout the year at 10-meter, 5-day resolution. And we find this really works really well, the Sentinel-2 data, but it has to be calibrated. You can't just use the raw data. So we've been calibrating this, looking at using what the standard has been for doing fieldwork is hemispherical photography. That used to be done with real expensive cameras with fisheye lenses. Now we're using a smartphone, spherical panoramic photography, which works really well and uses smartphones that are widely available. So that can be used for the overstory, and for the understory, then relatively low cost NDVI greenness meters we're using for that. And so we've been applying this in different areas, looking at urban LAI, and we find that, one, we need to do this adjustment. If we just use the Sentinel-2 data, we'd be off by about 26 to 64%. And so the calibration is important. Another interesting thing we've learned is that the understory in the urban area is higher than expected. So by doing this work, we've increased the LAI in the urban vegetated areas. But also learned that some larger fraction of that LAI than we thought is herbaceous, which has lower emissions. So that's resulted in an increase, but then also a decrease in emissions from that in the model. Now, of course, and that gets to the point that, well, just getting the amount of LAI, the total is not enough because the emissions of different plants are really different. And on a first order, we have to get the woody vegetation, the woody foliage versus the herbaceous foliage, because they are order of magnitude different in emissions. And we can do that with this imagery. And getting this tree cover, the global studies, again, they do well in large forested regions and natural landscapes, but not so well in managed landscapes. And Vinyak Sinha his group has done work in looking at this in at a variety of sites around the world, and found that the tree cover, which is missing from these data sets in urban agricultural landscapes, can cause differences in emissions of about a factor of 2. We've been looking at this in urban landscapes, where we can do a really good job down using half-meter data, using US NAIP imagery, which is available throughout the whole US. We use this segmentation, machine learning classification, and we can get a very high accuracy. And what we find here, I'm comparing it, this for the area around Irvine, the UCI campus. And on the top, we have our machine learning object-based classification. So this is very accurate, done at half-meter. The second image down is the 30-meter data that we've been using, part of the NLCD data set, which was 30-meter, and we thought, that was really good. But we find it's off by about a factor of 2. So it gets only about half of the trees. On the bottom is an ESA global data set. And you can see, as you compare that with that on the top, you can see the distribution's pretty good. It really gets where the trees are. Problem is, that database just says tree or no tree at 10 meter. And so by doing that, of course, even at 10 meter scale, everything's not tree or not tree. So we find that if we just assume it's all tree, it's going to overestimate. But by calibrating this global data set, we think that we can actually, calibrating it with our half-meter data, we think that we could use this global data set, because obviously to do the whole world at half-meter would be very time-consuming to do. Okay, so then the next step is we get what's woody vegetation, where the trees are, what's grass, LAI. But then even within the trees, there are huge differences. So we can have order of magnitude differences for isoprene, alpha-pinene, different compounds for different trees. So we need to know something about this. Now, what have models been doing? Models like Megan and BICE have in the past just assigned a single assumed tree speciation. So every city in the world has the same tree speciation, because what else could we do? We didn't have any data for doing anything different. So we've been working on this, though, and recognizing that we think it's not the case, obviously, that all cities have the same tree speciation and emission factors. And of course, there are a lot of tree inventories now. This is becoming more and more widely available, where cities will get tree inventories. Houston, Los Angeles, a lot of cities in the US have geolocated inventories of more than 1 million trees. The problem with this is that they're only the public trees. Some places in the world that might be, most of the trees. In the US, it tends to be only about 15% of the trees are the public trees. So we need to do more than that. One approach that the US Forest Service has been using are these intensive ground plot surveys, where they go in, do random plots. And that's been done in a few cities in the US, and that's nice for testing and assessing, but it doesn't cover all the cities. So we've been working on a lower cost approach called the virtual urban tree survey. And so this involves getting random points and then using Google Earth and Google Street View imagery to get tree species composition. And we combine this and assess it with these other data sets. So we randomly locate about 1,000 trees in an urban area, which means someone has to look at several thousand points. Then use multi-season Google Map imagery, and we can identify the trees using a tree key that has to be developed for each specific urban area. So we can't do this in one urban area and apply that key to other areas. But then we can do this and we get a tree species composition. And so here's a couple examples from Central Texas on top and East Texas on the bottom. And so starting with the top with Central Texas, the default that we've been using, where we had the same tree species composition for the whole US, was about the value we came up for Central Texas urban areas was about 20% higher than the default. So not so different. Then when we did East Texas, though, we found that it was 50% higher than the default value. So that's a substantial difference. And again, we also were able to show that we couldn't use the same key developed for Central Texas. We had to develop a different key for identifying those trees. So once we identify the trees, though, we, of course, have to have emission factors. We have a lot of emission factor data for some of the dominant trees in rural areas, but not so many for urban trees. And so, for example, in the Los Angeles area, we have, based on the tree inventories of public trees, there are about 800 urban tree species from 300 different genera. And many of those we don't have any emission data on. So Jesus Campos, a PhD student working with me, has been developing an approach for going through and getting emission factors for these species. And it's using a two-step approach where first does a rapid screening of most of the species and grouping them into high and low emitters, and then doing kind of our standard traditional high quality measurements on the most important species. But even with that and with getting all this emission data, of course, we need to use the emission data in the literature, and it's a big challenge. I've been looking for a long time and trying to figure out how to do this. I've looked at using some of the approaches that are used for anthropogenic emissions, for example, tailpipe emissions from cars, where they'll use things where they'll put a quality estimate on there, kind of an expert judgment. But it's difficult. It's difficult to take literature data, especially much of the emission factor data is back from the 70s and 80s. We didn't have as good technology as exists today. And so we've looked at kind of a simple quantitative way to do it, which hasn't been all that successful. What we're working on now is a emission factor encyclopedia. And so the idea for each tree genera would just have a description of what data exists, how we synthesize that data, come up with emission factor databases and recommended EFs. And of course, we would like this to be a community effort. Hope to have anyone interested in doing this. It's something that we obviously can't do on our own. But the idea is to kind of get this out there and have the community weigh in and develop something that is both more useful, but also kind of open, which is definitely not the case right now. So the emission factors in the BVOC emission models are completely opaque. They're a black box. You can't see where they come from. So this would make it more transparent. Okay, so I'm going to wrap this up just briefly talking a bit about these managed landscapes. They can be managed, right? Urban and agricultural areas. For example, in urban areas, and of course, tree planting programs, very big and a very good thing, right? We want to encourage this going on. A lot of advantages for cities for doing this. But as they increase the tree numbers, that's going to increase the BVOC emissions. But also, and that's sort of a direct thing that we could just say, okay, double the trees, we'll double the BVOC emissions. But we think that's not exactly the case. Most of the cities, we've been looking at several now, including Los Angeles, and the new trees that are being planted are very different than the existing trees. Fewer native trees in a lot of places are being planted. And what we've seen where we've looked is that the isoprene emissions of the new trees are higher than the isotrene emissions of the existing trees. Now, what about on a global scale? Tree planting programs, right? So in plantations, agricultural areas, we plant these trees. Is that possible that we could impact the BVOC emissions? Well, if we do some kind of back of the envelope calculations, it's been estimated that agricultural activity decreased global tree population from 5 trillion to 3 trillion trees, right? About a third of those are probably isoprene emitters, suggests that isoprene emitting trees went from 1.5 to 0.9 millions. So there's discussion about, planting 1 trillion trees, right? So what would happen? Well, if 70% were isoprene emitters, that could change total global isoprene emission by almost double it. So potential for a big impact there, especially if those trees have a higher isoprene emission than the natural environment. And there's some indication that we should expect different landscapes that have different isoprene emissions. And so we've known this for decades that, for example, early succession trees that grow in high light and can take high temperature, tend to be isoprene emitters. And of course, those are the trees that we tend to plant because they grow well. We plant them in urban areas and in other areas. So we have these modeling studies that look at land use change, and they tend to basically just say, well, as you have agriculture, you're just going to get rid of all of the emissions, all the BVOC emissions. That's kind of generally the idea. But actually in places where we look at where this is being done, and outside of places like putting in monoculture, wheat and rice and corn, and then you really do lower the BVOC emissions, although maybe not as much because there are some trees planted there. But in a lot of sustainable agricultural practices, they are planting a lot of trees, and they appear that they may be high emitters. A study in Kenya by Yang Liu and colleagues indicated that the agricultural areas, and this is based on ambient concentration measurements, had similar BVOC emissions to the forest, the native forest. We have some similar data in Colombia that would suggest that as well, that the agricultural emissions can be quite high. So I'll wrap this up. I guess the key points that I wanted to get across to you is that these BVOC emissions from managed landscapes can be significant. We really need to be working on these urban areas, and that's more important than getting these rural hotspots. But it's challenging. It's difficult to get these emissions in urban areas. It's also important because this landscape management can be changing isoprene emissions. And I want to acknowledge my group at UC Irvine, as well as the funding agencies that provided support for this. And with that, I'm going to end and go out of the slideshow here and stop share, I guess, there.